Hey guys, welcome back. Here are the notes for chapter 22. We're starting nucleic acids and protein synthesis. Whoops. And that is going to be something really big. I wish I was doing this in person because it's a really cool topic. So the first thing that we got to figure out is DNA versus RNA. All right. DNA stores genetic information of a particular organism. So that's where all of this like coding and genetic stuff comes from. RNA takes that information, translates it into the synthesis of proteins that your cells need. So when we talk about your eye color, your hair color, different physical attributes, that's the manifestation of your DNA from RNA making proteins in order to do that. Because your eye color is a certain color because of the proteins that RNA generates. So here, they're nucleic acids. They're unbranched polymers, which means they're just in a straight line, and they're repeating nucleotides. And we're going to go over that vocabulary. That's our big thing right here: RNA versus DNA. All right. So nucleic acids. Now you'll be able to read this, all right? But I've drawn some stuff over here. Nucleotides. They have three components. There's a monosaccharide, which is a sugar, a nitrogen-containing base. Those are our bases that everybody talks about, and a phosphate group. And that phosphate group is hugely important. DNA contains several million of these. RNA contains a couple thousand. So DNA is far larger, making it far more complex. A gene, when we talk about our genes, that's a portion of DNA responsible for making a particular protein. So your DNA, a chunk of it is responsible for keeping your eye color, or your hair color, whatever, your skeletal size, the amount of muscle you have, all that stuff. It's all written in that gene. Okay. So here you can see what we have going on here. These are our structure. This is our phosphate group over here. See? Our phosphate group right there. This is our monosaccharide. All right, now you know what monosaccharides here. I've drawn the Hawthorne projection here. There's our monosaccharide. And then this is our nitrogen-containing base. So that's all of the chunks that are put together for a nucleotide. And a nucleotide makes up RNA and DNA, so both. DNA is in the chromosomes, and we have 23 pairs. That 23 is extremely important, right? So we have all this information so far. It's pretty straightforward. The monosaccharide that we are going to talk about, all right, this is the R in RNA. It's called ribose. And you can see this is what ribose is. Ribose is. It's a monosaccharide. There is our Hawthorne projection. For DNA, it's deoxyribose or D2-deoxyribose. All right. And that DNA stands for the deoxyribose. All right, so that's what we have there. The only difference, which I think is very fascinating, is for ribose, for RNA, you have this OH group sitting right there. All right, but for DNA, that OH group is gone. Okay, so that's the only thing. For DNA, we've removed that OH group, and that gives us DNA. All right, so here, RNA, D-ribose, it's a monosaccharide unit. For DNA, it's D2-deoxyribose. There's no OH group on carbon number 2, hence D2-deoxyribose. We removed that OH group from the second carbon. Virtually, it's the same. What we are looking at is both of these are 5-membered rings. They're going to piece together very, very similarly, but there's this one little piece right there that makes a big difference in their functionality. And this is what I think is extremely fascinating.